So, good evening, everybody. Today, Antonina Dolgorukova will give a talk on the analysis of the single cell data from the Kaggle challenge on single cell perturbations. Thank you very much, Antonina, that you agreed to make this talk, and please start.
Hello, thank you for inviting me. And yeah, I'm going to share my public notebook, just elaborate a bit on the analysis I've done with R-Cerat package, which is a specific package designed for some quality assessment, analysis, visualization, and other explanation of single cell data. And in this competition, we have to work mostly with aggregated data, not single cell, but as an additional data set, we also have some single cell data. So it may be useful to get some knowledge on how it looks like and what we can find there. So this notebook is about examination of the quality of data for this additional data set with single cell data, and some quality assessment and identification of highly variable features. I will tell what does it mean here, and some clustering, and of course visualization with this dimensionality reduction techniques like UMAP, tSNE. And I'm not an expert in the field, so feel free, please, to correct me and maybe make some comments. I hope Alexander will tell if somebody chat something, so I can answer or hear your opinion on this. Okay, so as a little bit recall for those of you who not yet quite familiar with the competition, here we work with single cell data called a data train file, and it comprises single cell expression data after 24 hours of treatment with 144 compounds, one negative control, it's solvent, dimethyl sulfoxide, I think it's called, and two positive controls. All these data were collected from you see this number, 240,000, like so, human peripheral blood mononuclear cells, which were isolated from 3 healthy donors. So it's healthy condition here. And it's unusual for general Cerat analysis. We have gene expression data after treatment with drugs, not baseline. And let's see what we can find here. So here load the data. I just prepared sparse matrix which take much less memory, make it easy to work in Kaggle. Sparse matrix with cell IDs in columns, cell 1, cell 2, cell 4, and genes by rows. And these are counts. Zeros are hidden in sparse matrix. So also we load metadata from additional file. In this competition, we have this additional file with observational metadata in CSV, and we have to load it separately. So we have data on 3 donors, which cell belong to which donor. We have also 6 cell types. You can see all kind of cells we have here. We have data on what drug affected each particular cell. These are drugs names, and another column indicating whether the drug was controlled or not. And you can see that like one-third of the data comes from control. So much more cells were treated with control. We have only 3 controls, and this is this ratio. So we create surrogate object. I will skip these technical steps and do some standard pre-processing. It includes usually finding examination of mitochondrial and ribosomal contamination. The thing is, single cell data commonly consist of large numbers of reads from mitochondria, and this may indicate that these cells with most of the genes from mitochondrial genes just like dying. So, this gene go out and contaminate readings. So we may want to check the percentage of mitochondrial genes to be sure that cells are okay. Also, ribosomal genes. We're also interested to see the percentage of ribosomal genes because a little bit more challenging for me explanation why we do need this. Anyway, these genes, as I know, just not quite different from the others, so may somehow influence maybe all drug effect. If mostly these genes are affected, it may mean something for biologists, which I'm not. So the challenge is, at least with my background, to find a comprehensive list of all available mitochondrial genes. So far, I just managed to find some patterns. That means that gene with this pattern in its name is related to mitochondrial genes. So these are patterns for mitochondrial ribosomal proteins, and this is a component of some mitochondrial RNA processing endoribonuclei. I don't know how it's pronounced, sorry. Using these patterns, we can find all genes we have with these names. Not a lot here, like 86 mitochondrial genes at all in all that data set. And we can check what the percentage is. And here we see it's really, really low, from 0 to just 1.5%. Maybe it's good for, I think it's good for, meaning data quality here is okay, as I understand it.
How do you calculate?
Yeah.
So you just divided the total sum by the sum of these mitochondrial genes, or?
Yeah, this simple step which I did missed, creating zeroth object, it we come with this data frame with already calculated number of counts and number of features in each cell. Here is a cell, and we see in this cell there are 3,000 something counts. It's just sum of all counts in this cell of all genes. And in this particular cell, there are 1,700 different genes. And we already have pre-calculated this data, just creating zeroth object. And yeah, we divide number of mitochondrial genes in this cell by the number of overall number of genes. Here it is, and we get the percent of mitochondrial genes. Is it clear?
Okay, thank you.
Yeah. Next step is ribosomal genes. It's from the tutorial, third tutorial, yeah. It says that ribosomal genes may be very high represented and we can see what cells contain more ribosomal genes. They also say that ribosomal genes are a measure of the translational activity of the cell rather than the clearness of some quality of our readings. And here I found some patterns. It was almost a comprehensive search, so I used a huge database, and I found these four different patterns indicating that the gene with this pattern, its name is ribosomal. And we see much more ribosomal genes in this data set, more than mitochondrial. It's like 300. And the percentage is highly variable, from lesser than 1% to 77% of all genes are ribosomal in some cells. And similar data we add in our metadata table, percent of ribosomal genes calculated same way using counts, overall number of features, sorry, a number of genes meaning. And here just sorted just 5 with most large percent of ribosomal genes. Yeah. So now we know that the distribution of ribosomal genes is varying between cells. Maybe there is some meaningful difference between cell types, right? And because we have data after drug treatment, we also check if it is somehow related to drugs. Maybe some drugs, maybe these cells are treated with only these particular drugs or something like this, which may give us some knowledge about drugs effects. And we see median value of ribosomal genes by cell type. It varies also highly, I would say, it's a huge variation from 12 to 43, 44%. And myeloid cells have lesser amount of ribosomal genes usually. And CD4 plus cells have most, the largest percent of ribosomal genes. So this difference is vary across cell types. Why so? Maybe it's somehow related with our negative control or positive controls, which are positive control supposed to affect gene expression, and negative control had not to affect because it's like it's solvent, must have no effect. So maybe I had to make 3 groups actually here with negative, positive control, and false not control. No, but here in this representation we have no difference, so it's like almost similar. Now we check what about our compounds, and here just top 10 drugs with largest percent of ribosomal genes, and bottom 10 drugs with lowest percentage of ribosomal genes. And we see this CGP drug, cells treated with this drug have highest percentage of ribosomal genes. Interesting. If you know something about these drugs, maybe.
The 2 drugs which are positive control, are they here on the top or not?
Here, belinostat, which is positive control, you can see, and our negative control is here. And I don't remember the name of second, maybe. Don't you?
Me neither.
This one. No?
Well, maybe, yeah, maybe.
I think this is positive control, but maybe this one. I'm not sure.
Okay. Okay.
It's about ribosomal genes, yeah. So this is the same for mitochondrial. Not so informative because we already saw they very low represented. So I maybe just keep no difference again, and maybe not worth it to waste your time of looking at those drugs because percentage very little. So we can plot percent of ribosomal genes per cell type and see already what I already told. Myeloid cells have the lowest percent of ribosomal genes here, and almost equal mitochondrial genes. And these are our drugs. We can enlarge this picture. And we see a few quite different from others. So previously we saw in the table this one is this one huge bar is CGP 60 something drug have the largest percent of ribosomal genes, and here is it is very different from other, I guess. Maybe not all, but most, because most have just similar percent of ribosomal genes. Maybe this drug, maybe if you know something about this drug, you can explain this somehow. Would be interesting to hear. Same is about mitochondrial contamination, and here we see, just interesting for me, same drug, CGP 6474 with lowest percent of mitochondrial genes. So the same drug producing highest percent of ribosomal genes and lowest of mitochondrial. There are some biologists to explain this.
Have you checked that these examples, they are not in the list of these exceptions which have very few cells, single cells in these groups?
I think not.
Okay.
I think not. There was another. I saw. Yeah, I saw this one. Alvociclib was. Its effects were measured in a low amount of cells. I didn't found any relation actually to the number of cells. I did see. I examined it. Yeah.
So because mitochondrial genes, when they are high level, that means the cell is something like dying. It's very bad. So when it's low level, what should it mean? It's
maybe nothing here.
Something like it's a very healthy. Actually, I have not. For example, in the Python pipeline with the ScanPy, there is no mentioning of this ribosomal genes. So I haven't heard before that they have some people analyze ribosomal genes. So ribosomal genes should be about the production of the proteins, which is made on the ribosome. And maybe if, again, cell is in very healthy, good condition, it produces lots of proteins. But in this case, that is compatible. So it's, I mean, but on the other hand, we can expect that if there is this negative control, then it should happen there that cells are kind of in healthy condition, and we should see more ribosomal and less mitochondrial. But since it doesn't happen, then it's for me it's difficult to interpret.
Yeah, for me too. Maybe it's just coincidence. I don't know. Yeah. So two conclusions from the data from our analysis above is that myeloid cells have the lowest percent of ribosomal genes, and this drug looks like an outlier maybe. with cells treated with this drug have the largest percent of ribosomal genes and the lowest percent of mitochondrial. No explanation, sorry. Another thing to check in our single cell data, it's a percentage of largest gene. This means we will find gene that have maximal counts in each cell and calculate what percentage of all genes copies come from the single most observed genes. Meaning, so like you have three different counts of 3 genes and 100 counts from just one gene, meaning that something wrong with this cell. Also, this may indicate some biological context. It depends on what gene is. So we will check what genes account for most counts from cells and how often we can see them in the data. Here we calculate, I skip it, and overall the 36 largest gene means that these genes have its maximal expression in all cells. And we can see maybe some familiar genes, like this one, the nuclear gene, and this one maybe too, and this one is ribosomal, and this also. Also this, I can see, and this hemoglobin, right? Maybe you know something more about this, but by some reasons, these 36 genes represent most counts in all cells. So
most, that
yeah.
means how many?
Sorry?
Most counts, that means what percent of them?
Now we will see this below. I just wanted to a little bit explain my calculations because of we just count from which gene comes maximal count from a single cell, so we can get 2 genes with maximal counts, and since there are only 43 from our many cells, I just skipped. I just took the first one and skipped others. So, but I don't think this influence much on the overall statistics. So we just go with this. And here is the percentage represent counts. So sorry, single gene counts can represent from 1 to 79% of all counts in the cell. It's quite huge number here. And we add this new column with percent of largest gene and the name of the largest gene, and it's malat, I don't know. Here mostly this head of the data, and let's see how many cells have gene copies from a single most observed gene. More than 10% is in 2000s of sorry, I forgot to say, because this calculation require a lot of memory, I just took 10% sample here. So we now have 24,000 of cells, 10% of the data, and we go with this. Actually, in my local PC, I just checked the full calculation in full dataset, and it's quite similar distribution. So this analysis is representative in 10% of cells. And so where we're like more than 10% of all counts come from one gene, in 2,500 of cells from our 24,000, like this. And there are also 3 strange cells, maybe almost dead in our sample, but 3 is a very low number. Maybe don't mean anything here about the quality. So here I just sorted the data from largest percent of largest gene to lowest. And mostly we see this hemoglobin gene, this immunoglobulin gene, I guess, and this nuclear gene. I also don't have any explanation why I know about this one. It's nuclear, so it come out the cell when it is dying. So the large amount of this gene in some cell may indicate it's dying cell.
No, but I think this MALAT1, which is low non-coding RNA,
Yeah.
this is on the top level, I think, for most of the single cell data sets that I think typical. I'm not sure that it's related to dying of the cell.
Yeah,
Just
that's also
there
true.
is huge amount of this MALAT1 by some reason.
Yeah, sometimes. Yeah, in previous, in my analysis of previous open problems competition, we also saw this gene was most represented among all genes in our data set. So it's just common situation and maybe normal. So what number and percent of cell in which the expression of the largest gene is the highest respective to other genes? We have our 35 because we skip just one 35, okay, genes all are here, and this MALAT gene is the largest gene in most of the cell. And there is a huge gap. So previously we thought that this is a high variability, but obviously it's not. Just MALAT is outstanding here, and all others highest gene just maybe this one just equally represented in 1, 2, 4 cells. So it's just meaningless information here. Maybe these several genes may say something to you. I don't know, not sure.
This I don't remember. The other, this ribosomal RPL, which are 2 positions below, that's also typical that in single cell data we see MALAT and the ribosomal genes, they're typically on the top of the expressed genes. So that's not surprising.
Yeah.
But about FTA,
So, yeah.
I don't remember. Maybe it's just we need to check with the other data.
Yeah. And anyway, I don't see anything like extraordinary in these data yet. So all looks like quite fine.
Yeah, so some like hemoglobin, or for example, this HLA, this just because this is a blood cell, so it should be loss of hemoglobin. HLA, DRA, this is this histological immune histological complex, which is the should be present in the immune cells, I think.
Yeah,
that's one of the receptors.
Could be explanation. Yeah.
So
yeah, you're right. Also, we make the same check if whether the number of largest genes, the percentage of largest genes can be explained by control or drug treatment. It is not. So the distribution is quite similar. So cells treated with control and with our drugs have similar percentage of largest gene. So it's okay here. And this amount of larger gene is also quite equal between cell types, which is good. And not so variable among drugs, but this one outstanding here maybe, and now our CGP is something wrong with it. I don't know what exactly. Here we see this drug again, CGP, yeah, with almost one of the top with largest percentage of our largest genes. So, and this one, yeah, there are very few cells treated with the drugs in our data set. So maybe this is the reason why these cells maybe some these counts maybe some biased or just few, because mostly we have 1/300 of cells, and for this drug only 50 or something. I don't remember exact number, but it's lowest. And okay, so we can now just look at the pictures, what we have here in the data, and there are number of features, which is I can enlarge it a bit. Number of features, it's number of genes in each cell. So each dot is a cell. See how distributed a number of genes within each cell from, and we can already see here some cluster above main data with a very high amount of genes. So these cells have more genes than the main part of data. The same is true for number of counts. Number of counts, just summing up all counts from a cell. And here we see that for most of the data, this number of counts is somewhere like 5,000, right? And in some pattern of data, it is very high. Here also two violent plots for mitochondrial ribosomal percentages, and it is quite okay distributed. Here too, maybe these cells with lower ribosomal percentage somehow different from each other, but mostly it's well distribution, I think. And here our percent of largest gene. So we can see that just few cells, as I showed in the table, just few cells have very high percent of largest gene. So maybe we somehow can clean up the data before using in our models or in our further investigations, because these few cells are very, very different from each other, and there are just a few, so we'll not produce any strong influence of the analysis, but maybe worth it to remove them because they are quite different. So
the only remark I would say that I'm surprised that we have such a low percent of mitochondrial genes, because typically it should be in some tutorials they say less than 5% is okay, but in practice I have seen lots of examples where it can be 20% or something like that. So, and here we have below 1%. That means I don't know what does it mean. It means that a very, very, very excellent data, or I don't know. That's a little bit strange.
Yeah, it's actually really strange. Maybe it's related somehow to the methodology. They measured single cell data. I don't know. Yeah, usually we saw in previous competition we saw like 30%, and we was thinking about filtration of cells below some percent, maybe 10. Yeah, because we had.
Yeah, this is a standard recommendation for in the tutorials that we should filter out the cells with higher percent of mitochondrial genes. And something like less than 5%, for example. But here, everything is less than 5%. That's a bit surprising.
Very low. Yeah.
Yeah.
It's interesting to see some notes about pre-processing, how they achieved this, because it's quite unusual for single cell data. Yeah, that's right. So the next helpful plots are here with a number of features versus with anything we talked about before versus number of counts. So for example, here we see number of features, it's number of genes. I just didn't rename standard naming in Serot. So meaning number of different genes in cell versus number of gene copies. And here we see that there is main data with low number of features and lower than the median, I guess, number of counts here, this cluster, and very different part of data with very huge number of features, like in our violin plots, and very high number of counts. The same cluster we can see in all of these graphs. Percent of largest genes also prominent here in some pattern with, oh sorry, I said it wrong. Percent of largest genes, no correlation, but also this pattern of cells with a large number of counts here. And
that should be the same cluster we see.
I think so. Yeah, we will see further by cell type, called by cell type. Maybe we just can come to the next. Okay. Anyway, this person of ribosomal, yeah, and different cluster here, and strange picture like fingerprint of somebody with
that can be reasonable because look, if this is called mutually exclusive, that means if you have high percent of the mitochondrial genes, that means cells in something like bad conditions, and so number of counts should be low. So either one is big or another is big, and so you have something like a hyperbolics for this.
Here, I see. But this cluster
this is like a fingerprint. I think it's related to the number that it's a kind of integers, and maybe
yeah. So quite, yeah. And here again, no dependencies here in this cluster of cells. So we just will color these plots by cell type, right? We have different cell types, so it may be helpful here. So our myeloid cells and B cells, yellow and red, are which are cells in which we are mostly interested in, because we have to predict some drug effects in these two cell types, right? And we see that these strange clusters comes from mostly myeloid cells.
But B cells, I don't see the red color here.
Yeah, because very low amount and equally distributed in all plot, maybe.
Okay. And so you
here and here, I think, yeah.
Okay, and so it seems that these myeloid cells, they're kind of special, yes? So they have more counts.
More counts.
Yeah, something like
more genes inside each cell.
Yeah, but so the first picture is kind of natural, that number of counts should be very correlated with the total count. No.
Yeah.
But what is surprising that the cell types are that much different.
Yeah, cell types are separate in our data clusters.
Yeah,
so cell types
for me, it's
here,
unexpected.
It's very strange, yeah.
Well,
but you can ask me if you know how the data were processed and what we have here in this competition, you can recall that we have only few samples, few observations from myeloid and B cells because it's trained data. So we have only 15 drugs for B and myeloid cells. So all those dots may come from controls, right?
Then it's yeah. So if the cells are under the drugs, they probably have a kind of, I don't know, it's bad for them. They are kind of not in good condition, and so we see less counts, less everything, more mitochondria.
No, there's different controls, two positive controls which have to influence somehow expression, and one negative control which
yes, the positive control, these are the drugs which have a very strong effect. So that should be just opposite. This strong effect should almost kill the cells, and they should be okay. But have you colored this by the drugs?
Sorry, I just wanted to show you another picture. Oh, sorry, no, not this one. Maybe this one. Not this one. Maybe to clarify a bit why we have so many control, just important here, because we are doing single cell analysis, and as I told in the very beginning, almost 1/3 of data comes from control, right? So all we see in these plots, third of these are from control data, not from drugs, right? It's quite important to understand all patterns here. This just plotted the experimental setup. There were 6 plates. I have six plots here, and each plate consists of several columns, and in each plate, one column always was treated with 2 positive controls and 1 negative control. Almost the same column of each plate. All other wells in the plate were treated with different drugs. So to cover all 146 drugs, they needed 2 plates. So half here, 72, and 72 drugs here. All are different. And we have 3 donors. So because of this, we have overall 6 plates, 2 for each donor. And you can see why we have so many control data, because in each plate, 3 columns were treated only with controls, much more data from controls. So we go again to our Seurat analysis. Where are we? We were here, and think like this, think like that. Maybe we still wrong.
But have you colored by the drugs these things?
I tried, and it is just equally distributed. Very, very many colors here, and no patterns. I didn't show this picture. Nothing to show because of this. So I just wondered about this cluster. Yeah, we cannot see it because very little dots, right? So let's see numbers. And just filtered all cells with counts above 10,000 and number of genes above 3,000. So to separate this like this class this cluster here, and we see that if we just filter data like so, we see that most of the cells are indeed from myeloid cells. And there are a few other types. So there seems to be a distinct cluster with the highest number of counts, mainly represented by myeloid cells. Now we plot the same but color this by control. And we see that, as we already figured out that our differential cluster is mostly comes from control treated cells.
But
Yeah, and
what control? Is it positive or negative?
both. I just didn't. It didn't came to my mind to divide control to 3 groups. It's all
But
controls.
the sense is opposite. So the drugs with from the positive control, they should very strong effect. They should almost, I don't know, something like kill this. But negative control should do nothing, ideally, but
yeah. If I did so, we could already answer some questions, but I maybe partially answer it below another way. I just removed all controls, and we can see in a few minutes what changed. So yeah, maybe we'll add in this plot or dividing control to 3 groups or to 2 groups, positive and negative, and maybe it will be also catchy picture. I don't know. Anyway, so here nothing to say about these pictures. I just skip it. And now this is here I figured to filter out control cells. Yeah, just showing these plates, experimental setup, that I just remove these cells from these wells, right? And we just leave only drug-treated cells to see if they produce some patterns in our data. And what we do after, we do now we want to use other several tools to investigate our data, and this includes identification of highly variable features. Usually, it is usually for just, I don't know, to use these genes as features indeed in the model to predict some in multi-omics data to predict some proteins expressions, I don't know. But here, by finding highly variable features, we can see which genes are differentially affected by our drugs, which may be quite useful in the modeling to solve our competition. So our problem in this competition. So to do this part of analysis to find the most variable genes, we need first normalize the data, because now we have raw counts. And I will skip this part and show that the normalization was quite well, with just a few outliers here. So we now see what genes are most highly variable, meaning that some drugs affect, meaning that the drugs affect them very differently. So one drug lower expression, another drug make the expression higher. So this would be variable gene, high to low. So before this, we also have to scale the data and make dimensionality reduction. We do PCA here because there are too many data to process, and we need this reduction. Here, just I will skip these plots because I have very little to say about. We just see what is the real dimensionality of our data set, because after PCA, we have to choose a number of principal components to go with, and how to choose it. There are several ways, like elbow plot, showing standard deviation of principal components, and we see that maybe 4 principal components with high standard deviation, and after this it becomes like lower. And, also, another way is to compute percent of variance explained by each PC. It's like a way to choose how many principal components to take, to choose, to select. And here, I would say all of them does not explain well the variability. Anyway, it's okay here. This picture say not, maybe, I don't know what does it say actually. Maybe after 20 principal components, it became less informative. This one, I don't know. Another way is to construct a heat map, and the more the better we see borders between these clusters, the better this principal component explain variability in the data. So from this, I would say 4 is enough. 4 are good. These are blurry and not informative. Anyway, it's your choice. You can pick any number on your subjective opinion. There is no rule. So I proceed with, before I proceed, I just plot some pairs of principal components, PC1 versus PC2. We see where our different cluster of cells, which now doesn't come from control cells because we have no control now in the data. We just filtered out all control cells, and still we have, after the dimensionality reduction, we still have this cluster of just myeloid cells, very different from each other, from all other cells. I just put this additional picture to show no control here, all false. Still, yeah. And some other pictures, principal components colored by mitochondrial percentage, ribosomal percentage, maybe tip with highly contaminated. Sorry, this root, maybe, I don't know. Yeah, rather root. And our cluster from myeloid cells is very different by number of features and number of counts again. So still, after removal control, we see that myeloid cells are much different from all other cell types. Some more components. Still 4 and 5 still differentiate clusters. That's good. If we plot more components, I just tried, maybe component 10 versus 11, there would be like a cloud here, no clusters. So I go further with 4. With 10. I go further with 10. 10 also was okay. Maybe I just checked it with maybe not the best way. Anyway, now I do the analysis, finding neighbors and constructing clusters. It's to find what clusters our highly variable features produce. So again, we have highly variable genes, and these genes may differentiate somehow drugs or cell types. So we just see what clusters they form, and whether these clusters remind something, maybe, yeah. And here, oh, sorry, wrong picture, this one. And here, my clusters here remind very well clusters produced by cell types. So cluster analysis just differentiated this 11 cluster from myeloid cells, and here it is indeed one cell type, only one in this cluster. But if you look at maybe NK cells here, there are like 3 different clusters. So these NK cells consist of genes, of highly variable genes, which form 3 different clusters by their reaction to drugs. I would say like this. It's my explanation. And here, another interesting thing, we now see, we can now differentiate B cells too. This 10th cluster here, red, is B cells. So myeloid cells as well as B cells without any control are very different from other cell types. What our analysis just said to us. Any questions?
But also drugs we don't see. And so we see that this very elongation along practically PCA2, can we interpret it as something like, typically these things are interpreted as a number of counts. So we can, for example, just color this picture by the N genes or N counts.
Yeah, just saw it
And,
here. I think, yeah, here. It's here. Number of counts with highest counts here in our myeloid cells.
But yes, but this part elongated, which is not myeloid cells, but this yellow part, and we see the clear kind of something like direction here. Yes. So the question is how we can interpret that kind of what's going on here.
I don't know, maybe
percentage of the ribosomal genes, yeah.
Yeah, maybe this one, yeah, because the color is just go down here.
Yeah, interesting.
So yeah, another thing which we can do with SERAT is find biomarkers of each cluster, meaning that we can see what genes differentiate this cluster from all other clusters. And we can also compare two clusters, like myeloid versus B cells or myeloid versus NK cells. I still don't know how to use this in the competition, just for overall knowledge, I guess. For example, here I compare cluster 11. What genes are in this cluster most I don't know, indicative of this cluster, and also compare where it was, sorry, yeah, here compare one cluster versus all other clusters. Here clusters in x-axis, and just a few top genes which better differentiate these 11 cluster myeloid cells from other cells. And we can see that there is gene CTSS which is highly expressed in specifically in myeloid cells and much lower in all other cell types. This is what about this analysis. And this HLA hemoglobin, some blood gene also, and some other top genes here. And what else here is yeah, we did compare all clusters to each other, and we can draw this heat map with clusters, and we see and we plot just top 10 genes which differentiate these clusters. It's like maybe 30 genes overall for 11 clusters, and there are some patterns here. Each cluster is different from others with some specific genes. I don't know how clear it is. Maybe no, maybe because it's not quite clear for me.
No, but that's in a sense we constructed the genes such that these genes should differentiate the clusters, and that natural that we see this on the picture, that in this heat map kind of these genes and clusters, and we see that yes.
Yeah, like so. Yeah. Yeah. Yeah, like this. And I just also tried the same with dividing by cell type. And we see that our B cells and myeloid cells have a few genes, highly different from differentially expressed in these types compared to all other cell types, right? Also, a few pictures for the final, for the end of my talk. It's dimensionality reduction with UMAP. How do you say this? And tSNE, right? We can see almost same here pairs of plots by cell type and by clusters. And again, we see this 11 cluster and 10 B cells separately. And with this almost same, but differently depicted. Here are myeloid cells and B cells. Also, in this we have also two other different clusters that just separated, which are from NK and T, but I don't know how meaningful is it. For my opinion, PCA is better here, better to visualize cluster. I don't know. Maybe Alexander can comment somehow on this.
And do we have some interpretation that some clusters can be related to some drug classes, for example, or to something?
It would be great, yeah, if you could classify the drugs to groups, because if you just color these all three pictures with clusters by all 144 drugs, you just see equivalent distribution, no patterns.
That's strange a little bit, don't you think so?
Yeah, because I would expect, yeah, some differentiation, some groups by drugs, but we see only cells are different from each other, cell types specifically. So cell type is very, very important, I guess, in the prediction of drug perturbations. But how to use it?
No, in a sense we use it because we have these averages over the cell type. And when we add this, what I call priors, yes, and
yeah,
sometimes it's improve the quality of the prediction. So that in a sense that so yeah, these two quasi-constant solutions, one is average by the aggregate and average over the cell type, another is aggregate and average over the compound, and we add them to solutions, predictions, and sometimes we see the improvement of the score.
yeah, but for now, these features with target encoded, meaning averaged effect by cell and by drug is the best. Working comparing with all other features I tried here. But maybe we can figure out some better features for cells.
Yes.
So that's all from me. Maybe somebody want to ask or say something.
Thank you very much, Antonina, for your great talk. Everybody is welcome to questions, suggestions, or comments, whatever. You can ask the question by voice, so you can unmute yourself or write them in the chat. Up to you. You are welcome. Mikhail, any questions?
He can't unmute.
I have changed it, so.
Oh, thank you very much. Yes, thank you for your amount of work that you have done. Can you a little elaborate at the very start, first violent plots? What does what does it actually mean?
Even this one.
Yeah, like it distribution of not you counting number of features, and by number of features you mean I don't quite understand, because all genes are float numbers, right? Gene expression in our data set.
Here, number of features is number of genes.
Maybe
Like
we should mention that there are many genes which are just zero. So
okay, thank you very much. I have
Yeah.
a really brief understanding of the data set right now. Okay, it's much clearer now.
So this is the typical step in the tutorials, that we can count number of non-zero genes, yes? And
Yeah.
another, we can count just the total sum. They should be correlated, but there is some difference.
Okay, on the horizontal line we have
Nothing, just
Violin
stamp.
plot, just
okay. Thank you very much. That's all.
Yeah. So we have one number of genes. Dots are cells. How many genes in this cell? We have like 5,000 genes, different genes. Like in this cell, we have like 20,000, maybe 30,000 of counts if we sum all counts. Yeah. Thank you for the question.
That's all for me.
Any other questions, comments, suggestions? Okay. Thank you very much, Antonina, for the great talk.
Yeah, the main question is how to use this in the competition. I think maybe
yes.
people don't want to discuss it because it would be their solution. They can now go to their work and implement this in their analysis and get some profit in the leaderboard. Feel welcome.
There was just maybe today the topic on the discussion forum of this competition on Kaggle that so the person asked, does somebody use the single cell data, or everybody works with just the train set, which is just the pseudo bulk? And
I also saw.
Yeah,
I also saw one person.
He doesn't use a single cell. I think most of the people doesn't use. Most of the people use just the pseudo bulk.
Right, because this data is very, very, very strange. Having I just have made very sorry, a lot of different strange observations, like some data comes from just one cell sometimes, and there is no data for negative control sometimes, and it's not clear how they pre-process the data, or maybe I'm just stupid, couldn't figure out using all the descriptions, all the discussions, and I don't know, all I have still not clear how they subtracted the effects of negative control from to calculate aggregated data, and all of this not clear for me. And I did a huge work to investigate and examine all the data we have. I believe it's not clear at all for most of the people who just started and look at this and, oh my God, what do I have to do with this? Nothing is clear. I'm sorry if organizers listening, maybe you can answer some questions still unanswered in our topics in the discussion, and explain. So would have a good effect on our solutions. So you will benefit from this too, to explain us better. Yeah. And I saw also
this wasn't difficult even for organizers. At the early stage of the competition, they promised to release this code, but there was a delay because it's kind of non-trivial, all this lemma processing kind of.
Yeah, I saw also, I don't see it now, maybe I have to upload it. One guy said that he tried this approach, did you saw, predicting bug data plus lemma, and he just got inferior results compared to main data.
Yeah.
And also, yeah, I also saw from another guy who was saying that A data are somehow corrupted and can see there's some some mismatch he found.
Yes.
So maybe we actually cannot use this data because there are mistakes. I don't know.
You can try. If it's improve the score, then it's okay, and even if it's a mistake, it's useful. If it doesn't improve, then it's not okay.
It still doesn't mean it wouldn't work if it all was explained and correct.
Yeah. Yes. So yeah, you can try to aggregate something this data by counts, well, by subtypes, by drugs, and try to make some features from them. But I don't know.
But a lot of steps are not clear, just not clear, because in the data description they say that or somewhere else, maybe in Zoom meeting we had with organizers, they so our output is a significance of differential expression changes after drug treatment, right? So to get this, they somehow subtracted the effect of negative controls, they say. So it's our confidence on how drug affect cell, and their analysis account for solvent. But when I try to aggregate the data just by cell type and drug, I just found that there are a lot of observations with missing data for negative control. So like in this well, no data for negative control were collected. So how they could extract if there is no data, I don't know. Anyway, so yeah, thank you very much for your time and allowing me to share this little work. And
thank you very much.
Yeah.
Thank you very much. Okay, so we can stop recording just a moment.